So, in your own words, anybody can answer this. Tell me, what do you define as passive and active transport? Go ahead.
So like passive is like when a particle just goes through the membrane freely, and active is when like energy or a protein is required to move the particle.
Wow, they're really excellent definitions. Did you put that together? Did you read it on your own, or?
I had sixth grade science.
So they taught you about active and passive transport. That's excellent. All right, so it's a little more complicated than that, but I like what you're saying. I think you hit on the definition correctly, but does anybody else have an idea when they hear passive or active? And it's not something that I talked about last week, right? So unless you've had it in sixth grade, it's not something that you probably have heard about. Has anybody else heard? Can you think of an idea when you hear active and passive? Yeah.
Like even like I kind of did get like half of it, but I consider like the word active in the last question you had, and it said which one requires energy. So
That's it.
active, when you active, you're like using energy.
Very good. So you see, he saw the word energy, and he thought active probably has something to do with the use of energy. And so let's talk about what energy is, right? Let's remember that energy is what is required for movement. And there's all kinds of forms of energy, right? All different kinds, but as a general definition, it's what we need to move. What's needed to move is energy. All right? And so if we know that that's number one, and let me give you number two. Number two is everything, and we talked about this last week, we did. Everything is moving all the time. Right? We all know that. Is that right? Except for the exception of some things like, Kelvin, negative 273 degrees Kelvin, right? Then it's absolute zero, so then absolute zero, it stops moving, right? But everything is moving, even the table. We discussed that yesterday, right? There's always this like vibrational energy, not yesterday, but last week, right? Do you remember that conversation, or are we really going to? All right, could you just put it away, not have it out at all? It would really be wonderful. Could we think about what we talked about last week? I'm going to post it online tonight. I didn't do it because I was in the hospital with my mother all weekend. But what did we talk about last week? Yeah.
System about high and low, and then
We talked about high and low. That's true. Didn't we talk about things vibrating all the time? That atoms are always vibrating, always movement.
that's when I asked about somebody was do the same vibration or what like kill you.
That's right. You remember? I remember that too. So that's right. If we had the same, remember that he asked that question. Again, we're recording, so no names, please. Remember when he asked the question about what if something vibrates at the same energy? What would happen? And I said you could either cancel or they could end up ripping things apart. Right? We talked about that. We call it creating a standing wave or nullifying a wave. We talked about noise canceling headsets and how they work. Right? So everything is moving all the time is the point. And so everything's moving all the time. Energy is required for movement. So what does that mean? So what? By the way, you see these three dots? They mean therefore. They're used in geometry, so you might as well get used to what they mean. Yeah.
Does that mean that absolute zero is no energy?
That's right. Absolute zero is no energy, but that's not mentioned here. If number one is true and number two is true, what can you conclude? Yes. Everything has energy. So everything has energy. If one is true and two is true, then it has to be true that everything has energy, right? Does that make sense? Do you agree? Yeah. If it doesn't make sense, don't look up into the sun because you'll go blind. Don't look up into the ceiling when I'm trying to explain things to you. Listen, answer the question I'm asking because I'm talking to you, right? Do you understand this idea that everything has energy? Okay. It's a simple, do you understand Do you need to understand what kind of energy, whether it's potential or kinetic? Yeah, it's just, at some point you will, but it's not important. What you need to know is everything has energy, right? Okay. Now, when we're talking about active versus passive transport, what we're talking about is having extra energy. Having energy that, in other words, think about it. Not everything has the same amount of energy. Does that make sense? So if you have this remote control standing here, let's say it's on a stand and it's standing here, it has energy. We know that based on these two postulates in our conclusion. We know that this has energy. Now, if there was another remote control that was bouncing around the room, it would have more energy, would it not? If I pushed this, it would have extra energy, wouldn't it? Right? So energy, how much energy defines these words passive and active. So passive things do have energy, but they don't have extra energy. So we say active transport requires, here's the definition, you'll see it in a minute again, requires extra energy. Where passive doesn't require much, anything extra. Yeah.
So it's like, if an object can be absolute zero, how can everything have energy?
As I said, it's complicated, but let me say this. What I said is except for things that are absolute zero, and I don't know of anything that is at absolute zero. Absolute zero is a theoretical temperature. And it's absolute zero Kelvin, right? So it's a whole nother measurement of temperature, all right? So at absolute zero, it's a theoretical number. I don't know if we have anything at absolute zero, but it's also more physics. So see, that's not something that happens every day in biology, right? Biological systems don't have to worry about absolute zero. Does that make sense? When I see you staring off into space and then nodding when I ask a question, I know that you're just nodding to get me to shut up. It's like, come on, guys. You have to participate. All right, so let's read the paragraph. Would you like me to read it? Would someone like to read
No,
it?
you can read it first.
You go ahead and read it.
Now imagine someone sitting in a boat that is moving downstream with the current.
All right, let's stop there, because what happens is that people when they read, they try to read to get done with it, and they don't read to understand it. So let's read to understand it. Now imagine you're sitting in a boat. So everybody knows what a boat is that correct?
Yes.
Okay, because people say yes, but then some people, I can tell you this morning, some people didn't know what a boat was, or rather what the boat had. You know, the one that people got confused with is current.
It's just moving.
It's water that's moving, is correct. Anytime you have moving water, actually, it could be air currents, right? Anytime you have a fluid moving, we call it a current, right? Movement. A current is the direction of movement of fluid. Okay, good point. Anything that's moving in a direction, so any particles, including electricity, air, water, they all have currents, right? In fact, you might even be able to think of the idea of people moving in a direction. You ever see New York City on the sidewalk, everybody moving in one direction? You might say you're moving with the current if you're moving with them. So those are definitions that are a little bit off task. The idea is that we're talking about a fluid, but it could be particles moving in a direction. Do we got it? And that's called the current. So please understand it. Electricity, when you talk about current, electricity, it's when electrons are what? Moving. Very good. In a direction. All right. From what? From negative to positive. That's the movement of the electrons. So you're in a boat and you're moving with the current. What does that mean? You're getting a boat and you have to has anybody been in a boat before? On a river or a lake? Okay, so a lot of us have been in boats. Has everyone seen people in boats? Okay. I mean, we have to make sure, because some people haven't. Some people don't watch TV. They don't really think about it. Okay. Believe it or not, I've had things, you wouldn't believe some of the things that I've said that people didn't understand. So you're in a boat. You're on a stream. Let's assume it's not a rapid, it's a slow-moving stream. You're in a stream. You're moving with the current. You could take a nap, right? And you'll get to where you're going. You don't need to do anything, do you? You're just sitting in a boat, and is there energy involved? Yeah, there is, but you're not putting any of the energy in. There's no extra energy. All you have to do is sit there and let the current take you. Yeah.
It's like how like with the like with the dandelions and whatnot. All you gotta do is alright. It's like passive transport is like the parachute going down. Like you don't really need to do much.
That's right. It's anything falling. When you think of passive transport, think about things falling.
Yeah, but like
Yeah.
active is like when the dandelions gotta like get wind up under them to flow. Ish. You're kind of close to it. Let's see if we can keep moving with this analogy. You're almost there. So when we take a look at current, we're talking about things moving with the flow, like you're sitting in a boat and you're moving with the flow. You don't need to put any extra energy in. Now, what if you wanted to go upstream? What if you go a mile, you go take your nap, you wake up an hour later, and you're going slow, so it's 2 miles in an hour. You wake up, now you're 2 miles downstream. Are you going to get out the rowboat and row your way back upstream? Yeah? Okay. I'll pull out a motor, and I'll attach a motor to my boat, and I'll putt all the way upstream, because I don't want to row that much. That's a lot of rowing, and upstream is going to mean... So let's think about what up and downstream is. When we're talking about downstream or upstream, all water runs which way, up or downhill? Downhill. So when you're talking about downstream, what you're talking about is water flowing down a hill. And when it moves slowly, it's because the hill isn't very steep, right? When it moves quick, it's because you have a steep hill. A waterfall is the steepest of the hills, right? So if you're in your boat, and hopefully it's a little not as shallow, and you're taking your nap happily with your hat on so you don't get sunburned, and you're moving downstream with the river. When you're trying to go upstream, you're going to have to row uphill. That's a lot of what? Work. And what is it? What's the word I'm looking for that we've been talking about? A lot of energy. It's a lot of energy to go uphill. So for you to go the opposite direction takes a lot of energy, because you have to row upstream. So when you're going with the current, when you're going with the gradient, this is called the gradient, right? Because what is this? What is this part of the hill? What is this part of the hill versus this part of the hill? What is this part of the hill versus this? Give me a word. High, steep, high. This side's high or more elevated. What's this one over here? Low or less elevation. High to... So when you're going from high to low, you don't need it. It's passive. You don't need any extra energy. When you're going from low to high, now you're talking about active. You see the connection? Yeah,
That's right. That's right. Like going up and down the stairs. There's like a million different ways of looking at it, but the key is that when you're going from high to low in any kind of gradient, and a gradient means high to low, all right, then you're dealing with when you're going with the gradient, no extra energy is necessary. But when you're going against the gradient, you need to put energy into it. That's all there is to it, guys. All right, so now let's look at number 2, where it says, determine which action shows passive transport and which shows active transport. So which of these two, going with the stream, going downstream or going upstream, which of these two is passive transport? Yeah, going downstream. Which one of the two shows active transport? Why does active transport require energy? Because you're going against the concentration gradient. Or rather, in this case, you're going against the gradient. Okay? Of course, which requires energy, input energy, going upstream. That's right. Everybody good with that? Because if you don't understand that, it's going to be hard for you to understand the next. And by the way, I am going to stop at the end, right before, like 5 minutes before the end of class, and let's write down the vocabulary words that are going to be important that you make index cards for, that you study for the next exam. That's what I promised you, right? That we'd make sure we do that at the end of every
clown.
All right, cell transport. The cell must move different substances into and out of the cell, right? So we need to be able to transport things. We understand that, right? Yes, please, someone turn the lights off.
Now these are the notes.
Well, that beginning part was important. Don't blow that off. That was a real conceptual issue there. All right, but remember, I posted this on the website, and I'll be uploading this to the YouTube channel, so there shouldn't be any issues.
What is the channel?
I'll give you a whole flyer on it. All right, so
You get paid for it?
no, it is not monetized because I just don't. I don't think that's right. So I do it for, it's not, let's stop talking about it, darling. Let's get to focusing. The cell must move different substances into and out of the cell. We all know that, right? We talked about that. Those of you that were absent, please watch last week's videos. And most of you that were absent, there's a couple of you, you probably understand the idea that things have to go into and out of the cell. This happens in many ways, and we're going to talk about all these ways very quickly now. Sometimes the cell has energy to use, and sometimes they don't have to use energy, right? Sometimes it has to use energy, sometimes it doesn't have to use energy. What are we going to call it when it uses energy? And what are we going to call it when it does not use energy? All right, so we got that. All right. Discuss the different methods of cell transport. All right, so that's those are the two things we're going to do today. We're going to look at the different variety of ways things go into and out of the cell, and then we'll talk about how to classify them. Another jacket. Oh my God, everybody has this jacket, the same style. All right, moving on. Notes. So here you have the cell must move in different substances in and out of the cell. We know that, right? Cell has to move stuff in and out. We talked about that, right? I mean, that's required. If you're going to have something that's alive, it has to be able to get, food in, right? And it has to be able to get what? Oxygen in if it's an animal, and if it's a plant, either way. It's going to have to get what? Nutrients, vitamins, right? It's going to have to receive signals. And so in order to get things in, of course, stuff is going to have to leave too, isn't it? So stuff's going to have to leave. The stuff that it's producing, if it's a pancreatic cell, it's producing insulin, right? So insulin has to get out of the cell somehow so that it can go into the blood. And of course, it has to get rid of carbon dioxide if it's an animal cell, oxygen if it's a plant cell. But it has to get rid of carbon dioxide, and it has to allow some other things out too, like waste. Every cell produces waste. You pee it out all the time in the form of urea. In the case of animals, plants have different kinds of substances, but we have urea. It's yellow, it's smelly, it smells a little bit like ammonia because in ammonia, urea is another form. It's ammonia that's been changed into something that's non-toxic to our system. Yeah. Yeah.
One is, like, when you said something about pee and go, well, what about, like, when you drink a lot of water? How come you see through dish?
It's clear. It's diluted. It's diluted versus. That's right. Someone said it's more diluted, and so that goes back to that concentration of solute. So the more water you have, versus urea, the less concentrated your urine. That's why. Question?
Isn't it when it's dark yellow that means you're dehydrated or something like that?
That tells you you're dehydrated because you have less water, but you have the same amount of waste, right? Because you're alive, right? And you're not acting you're still moving, you're still using energy, you're still metabolizing. Remember that word metabolism? You're still metabolizing, so you're still producing waste, but you don't have enough water to dilute it, so you have really concentrated urine. And concentrated urine is very yellow, very dark, even amber, and it smells really, really strongly, right? Because it's the smell of urea. Now, we don't have time to really keep on with these, so I'm going to ask you to write these questions down, and we'll pause, and I'll ask people to state their questions, and we'll discuss them at the fast break, okay? So discuss the different methods of cell transport. These are kind of our goals for today, right? So we're going to talk about the variety of ways we transport things in and out of the cell. See all the reasons. These are just some of the reasons that we have to transport things across the cell. There's many other reasons. And then we have to talk about how we do that. And that's what we're going to do today. All right, let's move. Let's take a look at this passive transport. And passive transport really comes down to one word, and that's called diffusion. And we talked a lot about that yesterday, right? Or last week, you know what I mean. So the last time we met. So we talk about substances moving what? Down a concentration gradient. And when we talk about concentration gradient, a lot of people don't understand what gradient means. Gradient means whenever you have something that's a lot on one side and less than the other. So when you have a gradient on a hill is a gradient, isn't it? And what people will ask you is, what is the hill's grade? And what they don't mean is they have an A, B, C, D, or F, right? They don't mean that. They want to know how steep it is. The grade is how steep a hill is. Now, another usage of the word gradient is in art, where you have stuff like this when you're doing what's called cross-hatching, and you have this section here, right? And then as you move down the gradient, what happens? It gets lighter, right? And that's also a gradient. They're called shades. There's a shade difference, isn't there? So you have high, we call this high, right? Or in art you would call it dark, right? All the way darkest to what? To lightest or lowest, right? So high to low is a gradient. So anytime you're going from high to low, we call it a gradient. I've used that word a lot, haven't I? Gradient, gradient, gradient. Guess what? It's one of those important vocabulary words, right? So concentration gradient, substance, diffusion. All right. So concentration gradient is the area where you have high to low. Anytime you have a separation from high concentration and low concentration, you will find it. You'll find you have a concentration gradient. Now, this PowerPoint is already posted on your website. So if you need something, you missed something, you can go view that. Also tonight, I will be uploading last week's lessons and today's lesson. And I'll be posting it on your webpage as well. So you can go to your webpage, look at second quarter, second marking period, and you can access these notes. All right, equilibrium. What's an equilibrium? We talked about dynamic equilibrium last week.
It's like, I remember when you said something about blood, it's like the reactants and the reactors, like, happen at the same time, so you can't know which is which.
Okay, that's I'm not sure.
I remember in that one equal.
Yuri, they want you in the office. So what's happening here is when we're talking about diffusion, you got to remember that particles are moving all the time. And when we're talking about fluids, water in your blood, for instance, or water in a beaker, and this week's lab, we're going to have a lab this week. Tomorrow we're going to do some work on the computer. Then Wednesday, and we're going to start putting our eggs in the acid to dissolve the eggshells. And then we'll play with some bubbles this week as well. So you're going to create your little your membranes with a pore in it. But you have an opening in a membrane. If this is the membrane, right? This is the membrane here. Remember that in a fluid, the good thing about a fluid, when you have like blood is a fluid, right? A liquid or gas. Gases are also a fluid. When we have a fluid, what's cool is that the particles can move freely. So these particles are moving around in their space, are they not? We all understand that they're bouncing. Do we understand that these particles are not standing still, they're moving all over the place in here? Mm-hmm. Do you agree? And do you agree that these are also moving all over the place on this side? Again, I have people staring off into space. I don't know what's going on today. Mondays are just the worst. All right, so if these particles are both moving on with each other, what happens if I only have a few particles on one side and have a lot more on the other? What do you think is going to end up happening is this is high and this is low? What? It's going to go from high to low, high to low, always high to low. Why? Because these are all bouncing around, and what are the odds that one of these is going to go through this hole? Are they greater or less than one of these going the opposite direction? Greater. So even though you are going to get movement in both directions, can I use a big arrow to represent the fact that this is moving a larger direction towards the right than to the left? A larger amount of these are moving to the right rather than the left? Is that okay? Okay. So the reason you get a greater movement is because on the left side you have more particles because they're high. On the left you have less particles because they're low. Don't confuse it because it's not always going to be high on the left and low on the right. Sometimes you have to somehow figure out which side is high and which side is low. Is that understood? Okay. So you're going to get most of the movement moving towards the left. All right, excuse me. So that's called diffusion, and so some substances move through a lipid bilayer. What substances are going to move through a lipid bilayer without proteins? What substances do passive transport? Oh, by the way, what is equilibrium? I should draw it for you. I apologize I didn't draw that. I just talked about diffusion. Equilibrium is this, when both sides are what? Perfectly balanced. You have 5 on one side and 5 on the other. So when they're even, when they're equal, equilibrium, when they're equal. And do things stop moving then? No, you have a particle, every one particle that moves this way, what do you get? One moving the opposite direction, so the arrows are the equal size, right? You'll notice here the arrows were pointing mostly to what? Mostly to the right. And here, equal. And when they are equal, when they're moving equally in both directions, we can't tell in which direction they're moving. You'll understand better when we do the lab, but I think you'll get more of it. The feedback I got from you guys is, can we go over the stuff first and then do the lab? And so that's what we're doing, okay? So this movement is called dynamic equilibrium. Dynamic equilibrium, right? Why is it dynamic? Anybody that remembers from last week? What does the word dynamic mean? So it sounds like a vocabulary word that we should write down, right? Because you need to know it. What does dynamic means? Moving. So this dynamic means it's always moving. And what does equilibrium mean? Equal. So dynamic, it's moving in both directions, but they're equal. We call it dynamic equilibrium. And when you get mostly to the right, this is diffusion, right? Or moving mostly in one direction. Yes. All right, remember we're writing our questions down, but we're moving on. Some substances diffuse through the lipid bilayer. What is a lipid bilayer allowed to move through it without any problems? Right, oxygen's an example, so I'll write it down. Oxygen gas. What's the other gas I told you better know? Carbon dioxide, you better know it. Oxygen gas and carbon dioxide gas. These two gases, you see the little G for the state of matter it's in, right? And at room temperature, 1 atmosphere pressure, what we call STP, oxygen and carbon dioxide are gases. Why are things gases at room temperature at 1 atmospheric pressure? Why are they a gas? Because they're what? There's two things I told you they are, yeah.
You said something about the particles being, I think you said the particles being spaced apart, not like forced together, so it's not a solid.
Right, they're not being attracted to each other, so they're not a solid. And the reason they're not attracted to each other is because they're nonpolar. Right, they don't have the opposite charge thing that we talked about when you have a polar molecule. So polar molecules tend to be, oxygen, water and CO2 are about the same size, and yet water is liquid at room temperature, while CO2 is gas at room temperature. Why? Because CO2 is nonpolar, where water is polar. The water molecules, they have cohesion, right? They attract each other. Remember those words, cohesion and adhesion? They attract each other. So because they're nonpolar and they're small, right? Small and nonpolar. Fats are also nonpolar, but why are they not gas at room temperature? Are they small? No, they're big. So things that are small and nonpolar, they're gases at room temperature. So gases tend to what? Move. The gas is like, so small and nonpolar flow through the membrane both ways, no problem. See, there's a double-headed arrow there? They move right through the membrane. They diffuse. So the things that diffuse through the lipid bilayer, very important that you know this, put it on an index card, tattoo it on your eyelids, whatever works, is the things that diffuse through the bilayer are small and they're nonpolar. That's it. Are we good with this? Everything else, things that aren't small and nonpolar, what do they need? They need proteins. They need transport proteins. They need transport proteins. They need some way through it. They need a door. Are we good? All right, so let's go on. So cell membranes are selectively permeable. What does selective mean? That's right. Excellent. They choose. Selectively means, and you better write it down so you know what it is. Selectively means choosing. So how does a membrane, remember a membrane is nothing but a bunch of phospholipids with their heads out towards the outside and heads towards the inside, and then they have fatty acid tails, right? So you have a phosphate and two tails. You remember that? So you have that. So that's a phospholipid bilayer. It's a bilayer. Why do we call it a phospholipid bilayer? Why bilayer? That's right. Bilayer, two layers. And they're phospholipids. All right, so these phospholipid bilayers, they tend to be barriers. Right? They're walls that protect the inside from the outside, to separate the inside from the outside. So nonpolar interior of the lipid bilayer, that's this part here. So this side's nonpolar. We went through this. We all know this, right? I don't have to go through this again, do I? No. Okay, could we answer so I feel confident? Because you make me feel... I feel like I have a lack of confidence when I don't hear you returning. I feel like I'm a person having a conversation with one other person in the room. I'd like to have a conversation with everyone in the room. All right? Please. Including you. And you. And you. Although you I have had some conversation with. All right. These substances are prevented from diffusing across the membrane. What stops things from, so this bilayer stops things from crossing the membrane. And so here's a question for you. If I have two cells, let's say I had two cells, cell 1 and cell 2, all right? They both have phospholipid bilayers, right? We agree? Why is it molecule A can easily get through in both directions through 1? You probably know the answer. And here, in cell 2, it can't get through. Why is it that in cell 2 it can't get through, and cell 1 it can get through? Same creature, same tissues. I'm going to ask someone else because I want to hear from somebody else. Go ahead. No, it's the same molecule, so it's the same quality. It doesn't change. So, but one cell lets it through, the other cell doesn't. Somebody else give me an idea? I see the same hand. I don't mind. I want to hear from you, but I really would like to hear from someone else. Go ahead. Don't worry, just say your thing. Just say it. Don't. It says what now? I say if this molecule was trying to get into the cell, both cells, and it can get into one, but it can't get into the other. Why? What's the difference between two cells? So this is your heart cell. This is your heart cell, a cell from your heart, and this one's a cell from your leg muscle. Well, if it was small and nonpolar, it would get through both lipid layers, right? So there's no difference between the bilipid layers, is there? So what's the well, there is a difference in the bilipid layer, but it's not the phospholipids. They both are made of phospholipids, right? So what's the difference between 1 and 2? Anyone else? Okay, yeah, you want to guess? Just make a guess. It's okay if you're wrong. Who cares? Well, they're both selective. One's selecting to allow it through. This one says yes, and this one says no, right? So this membrane says yes, and this membrane says no. Why? What's the difference between the two membranes? It's not the phospholipids, because they're both phospholipids. The proteins. I know you had it. I want other people to see what I'm saying. I want other people to volunteer. Yes, you may. Yes, you may. I want you to participate, though, but... All right, so yes, the proteins. This cell has proteins that allows A through, and this one doesn't have those proteins. You see? So, by the way, this is incredibly important that you understand. This is one of those subtle points that people miss. This is how the DNA controls what each cell is. That's how you make a heart cell versus a leg muscle cell. It's the difference in the proteins. So here you have protein A. Let's call it protein 1.2 is in the membrane. Here there's no protein 1.2. What does protein 1.2 do? Right, but what is one of the differences? This is obviously all made up. I hope we know that. So what is the difference? What do you think protein 1.2 does? Create doors, selective doors. Protein 1.2 allows what in? A and only A. If there's no protein 1.2, then A doesn't come in, does it? If 1.2 is there, then it comes in. And that's how your DNA, that's how your nucleus controls what goes on inside that cell. That's it. When we call the nucleus the brain of the cells, that's what we mean. Because the genes that are in that, which genes are turned on, determines which proteins go in that membrane. Which determines what's allowed in and what isn't. Does that make sense to everybody? Yes or no? Okay. Say sure, I hope it does. We're going to move on. You'll see other examples, but I can almost guarantee you're going to get questions. Well, let me rephrase that. I am going to guarantee you're going to get questions about it on the test. And we're scheduled to have our test on Friday. So, yes, a quick question. We don't have time for any complicated ones. Go. Yeah, and go get some or whatever you got to do. All right. Very small nonpolar molecules can go in and out, no problem, but the large ones are controlled by the proteins. All right, let's go ahead and look at the next slide. Here we have simple diffusion. And with simple diffusion, the kinds of things that get through are what? Things like oxygen and CO2, which are what? Small and what? Nonpolar. They just go right, they are gases, you're right. And they tend to be gases. Not everything that's small and nonpolar is a gas, but most things are. They're small and nonpolar. And small and nonpolar materials, and of course gas, liquid, or when we say are a gas, we mean at room temperature, right? Because we could turn almost anything into a gas, right? Does that make sense? All right, we're having some feedback on this system. Wish I could. Hello, 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 hello. I guess that's, turn all the rest of these down, maybe that would work. Is that, am I still on? Hello, 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 hello. Okay, that's good. Hopefully that worked. Nope, still getting feedback. All right, whatever. All right, so let's go through our steps, the questions we would normally ask. Question. All right, let's assume that these particles, I'm going to write it down, you should too, and you should sketch something like this out. These particles, I don't care if you draw spheres or dots, it doesn't matter, are small and nonpolar. That's number one. That's our postulate number one, if that's true. And then the next thing you have to ask yourself, somebody tells you this, you have to be told this in a paragraph, or somehow you have to be able to identify if it's small, not polar. The second thing you have to ask is, does it cross the membrane then? If they're small, not polar? Yes, then they do cross the membrane, so yes, we got that done. Then the next question is, which side's high? Which side's high? Right. Which is high? Which is low? So this is the left and this is the right, right? Is that correct? Everybody okay? This is high and this is low. Everybody okay with that? What is this thing? What do you see here? What is this thing? Some people didn't know that first period. What is that?
The membrane.
The membrane. Very good. It's real simple. It's the membrane. So it's the phospholipid membrane. That's it. That's all that is. So you got high and low phospholipid membrane. This is two polar heads are blue, in this case, and yellow. And we've been using the same color code all throughout, right? All right, so what direction are these things going to move? Left to right, high to low is correct. And so we're ready to go. That's called simple diffusion. That's all there is to it. I gave you that word last week. Those two words together, simple diffusion. Now, let's go on to the next slide. I want to reiterate, when we're talking about diffusion, what we mean is, in this example, what we mean is, and here in this example, it's a liquid, right? And we'll be talking about liquids mostly, but gases also diffuse. For instance, if you're in a corner of the room over there, and you put some perfume on, and it's one of those real strong perfumes, we will smell it on this side of the room, eventually, won't we? Because it's going to go from high concentration all the way to the other side of the room to this one. Another great example, although some people find it disgusting, I think it's a great comedic, a fart, yeah. If you fart on that side of the room, where the people closest to you are going to smell it the strongest, right? But over here in this side of the room, we'll smell it too eventually, if it's really stinky. Is that right? Because you don't have to be next to someone to smell it, right? And you've all seen bloodhounds, right? When people are chasing people in the movies, they bring out the hounds, they'll say, bring out the hounds, and it goes, and they're chasing people through the woods, and et cetera. Well, you ever see those police dogs? They'll take, like, somebody's missing, they'll take somebody's shirt and make the dog smell it, and then the dog will follow the trail. What trail are they following? The smell from what? From high to low. Well, actually, they're going to start usually at what? At the low. They will smell the low because they're more sensitive, and they'll follow the trail wherever it's stronger. As it gets stronger and stronger, they're going to follow that trail. They're like when you're trying to figure out, is that sound from the right or the left, right? Where's that ambulance? Is it behind us, in front of us? You listen, right? And you can tell depending on where it's loudest. Well, the dog is doing the same thing. It's sniffing around, and wherever it's strongest, that's the direction he moves. That's how things track you using smell. So that's high to low, and in gases as well as in liquids. So here you see an example of sugar. And with sugar, you have this cube. It's a solid. You've all seen sugar cubes, right? It would be the same thing if you put a tablespoon of sugar in the water. When you put a tablespoon of sugar in the water, or a sugar cube in the water, is part of the water sweet after you mix it up? Is part of the water sweet and the other part's regular water? No, it's all sweet, right? Why is it all sweet? Because the molecules of sugar do what? Dissolve, and they evenly distribute, don't they? That's right. It is partly due to hydrogen bonding because it's actually more polar dissolves polar, like dissolves like. So dissolving is when things surround each other and they turn into a fluid. The reason they go into every spot that they can inside this liquid is just the average kinetic energy of the molecule is going to bounce it around. And like I said, if all of us were sugar molecules, I know some of us are sweet and some aren't, but if we were all sweet sugar molecules and we were all packed in this corner and we started to shove each other around, what's going to happen eventually? Well, hopefully someone's going to move, right? another example would be, what if we were all on a really slippery surface? We're talking about a basketball court with grease on it. Really super slippery, and we're just barely standing still. And we're all close together. What happens if we're all moving around all the time? What's going to happen? We're going to fall, but what's going to happen if we keep moving even after we fall? What's going to happen to us? We're going to spread out, aren't we? And the closer we are, the faster we're going to spread out. Does that make sense? Well, that's the same thing that's happening here. These are all bunched up together, and you put them in the liquid, and they're moving. Now they're able to, once they're in the liquid, they can move around. When they were solid, they couldn't move around, could they? But now they're in the liquid, they can move around. And once they move around, they're moving around everywhere, every which place they can, because they're always constantly moving. Remember that. They're always moving around in this liquid. And so they're going to move as far away from each other as they possibly can. And that's why they evenly distribute. Does that make sense? So this is, of course, equilibrium. This is when you've reached equilibrium. This is where it was not at equilibrium. This is high, and this is low. So if you were going to draw an arrow, you would draw an arrow from high to low, right? But here, they're all evenly distributed. All right, so let's go on to the next slide. So here you have this highly concentrated molecule. So you see the arrows? Again, molecules are moving around all the time. Even in a solid. The difference is in a solid, they can't go very far because they're all bunched together. So they just vibrate. But you dissolve them in a liquid, now they have freedom of movement. So they can move around even faster and further. And then if you put them in a gas form, now they're really moving around because now they'll just keep going until they bump into something. So here they are bumping around each other, which is, by the way, why a balloon inflates. Why does a balloon inflate? Why does it stay inflated after you fill it with and tie off the balloon? What are the particles of gas doing to the side of the balloon? They're bouncing it off. They're pushing it, they're constantly bouncing it off. That's why the balloon inflates and stays inflated. Yeah.
So how come like helium balloons they slowly go out?
Oh, it's a good question. All balloons will do that. Why? Because they leak. There's leaks in them. There are some balloons that we have, weather balloons, that'll stay up in the air for months. In fact, in Puerto Rico right now, the hurricane knocked down all the cell towers. Google is putting up cell towers in helium balloons up in the atmosphere, and they're using those to bounce off cell phone signals off those because there's no towers. Yeah, I thought that was bright too. So these are all bouncing around in this space. You see it? Now, when you open a door, have you ever seen, what's a good example of this, I think, is a bingo. You ever see a bingo box where the balls are all roaming around, or lottery? You see the lottery? And then as soon as they open a door, what happens to one of those balls? They fall out. So that's what's happening when it's closed, when they can't get through, they're all bouncing around in here. Once you open that door, though, what happens? They're going to come out, and they're going to keep bouncing around. So they're going to come hit this and then bounce back, but there's more over here, so they keep going until they're what? In equilibrium. They notice that they're all moving in every direction. I don't like the fact that they drew it so that all the arrows are coming in. They should have drawn one or two going back this way. Because for every one that comes in, what must happen if it's a dynamic equilibrium? One goes up. Otherwise, the concentration would change. Does that make sense? Okay, so let's look at what it really looks like. And we're going to do this in the classroom, in lab, but I just wanted you to see what to expect. So when you sit there, you see this thick layer, they haven't mixed yet, have they? But they're still jumping around, they're moving around in there, just like these, and so are these. Over time, what do these start to do? They start moving around, they start spreading out. They start spreading out, just like us if we were in a corner of the room on a slippery basketball floor. If we were all bunched together moving around, we'd end up spreading out. And that's what these are doing, they're spreading out. And that's that represents number 2. Number 3 is when they've spread out a lot and they've come to an equilibrium. This is not quite there yet. What would you expect when this is all at equilibrium? What would you expect? You see, this was dark blue or black. This is yellow. When they start mixing, what color is it? Green. That's because blue and yellow make green, right? When they mix together, they can make green. What do you think? This is spread out, so it's not a little thicker. The layer's thicker. This blue-green layer is also thicker, right? What's going to happen when these two are completely mixed? They'll all be what color? Green, right? The black and yellow mixed together, they'll all be one green color all the way through. So that's what we call homogeneous solution. In other words, this is where we would reach equilibrium. We're not quite there yet in this picture. Let's go on and move on to the next slide. All right, so now something that one of the students said earlier was really caught me, because I know he said that he had covered it in sixth grade, and it was really great that he knew that the difference between active and passive is that active, you need energy. That's absolutely true. Active transport, you need energy. And it's also true that for active transport, you need a protein. You need a protein that allows you to cross the membrane and allows you to go from from low to high. That's the key. That's for active. But this is still passive. So I want to make sure you understand that. Some forms of passive transport, this is where it gets a little tricky. Some forms of passive transport require proteins. Facilitated diffusion is the type of passive transport that requires proteins. When I told you that proteins in the channel protein was an example, when I told you what does it do, what does protein do to move things across the membrane? What does it do? There's a word. Give me a word. Give me a verb. Or an adverb in this case. It does what? Say again? Bounce. Not quite. So protein is used to do what across the membrane? To move it, to help move it, right? Didn't I say help move it last week? I think so. I made a point too, because mentally in my notes I wanted to make sure I used the word help a lot. So proteins help things cross the membrane. Is that right? Some proteins. Transport proteins help things cross the membrane. Another word for help. Can somebody give me another word for help? Assistance. All right, assistance. If you got, okay, that's fine. But have you ever heard of the word facilitated? Well, guess what that means? To help. So, facilitated, write it down, because this is a word you're going to see a lot in biology, and it's a word you'll see a lot in English. Facilitating means help. It's a helper. So, facilitated diffusion is diffusion that requires proteins or help, right? Facilitated diffusion requires help or proteins. And that's how you move these ions and polar molecules across the membrane. Remember, they won't cross the membrane through just simple diffusion. They need help. And an example, you have an example that you have on those models up there, don't you?
Yes, I'm done.
Oh, is it time to go? Oh, Lord Jesus. All right, get out of here. Notes are online. Here's our channel protein, right? You made it. You made it three-dimensionally. You put it in the membrane. You made the membrane, right? So this is not new to you. Ions, sugars, amino acids can diffuse with the help of proteins. So you're eating an apple, crunch, chomp, chomp, chomp. You swallow it, you break it down in your stomach a little more, right? It gets into your intestines, out here in your intestines. Here are the cells. Here's a cell in your intestine. How does it get the amino acids that are out here, or it might be a sugar, simple sugar that's out here? How's it going to have a big sugar molecule that's polar out here in the big and polar into the cell? You need a what?
Protein.
You need a protein, something that's like this. So this channel protein just lets the ions through, potassium and sodium. When you drink Gatorade, how do you get the sodium and potassium into your body? It's not by drinking. Drinking's only the first step. The next step is you get that from your intestine, which is still outside your body, into your body by these channel proteins. We'll talk about that later. I tried to tell you that when we did anatomy, but we'll see. Proteins, sometimes called pores, often serve as tunnels through the lipid bilayer. We talked about this, didn't we? Who said no?
Yes.
Don't say no, trying to push my buttons this morning. They act as tunnels. We talked about channel proteins acting like tunnels. Each channel allows the diffusion of specific substances. So this channel will allow sugar through. Another protein might let something else through, okay? Only sodium, for instance, in this example, only sodium ions can pass through sodium channels. Potassium couldn't go through the same channel. Does that make sense? Unless it's specifically designed to do that. So these proteins are very specific. That's what we mean by control. That's what we mean by semi-permeable. That's what we mean by selectively permeable. Select That's how you select. Your body selects what the cell allows into itself and what it doesn't by choosing what? The protein. What protein am I putting in my membrane? So remember the whole ribosomes, the whole DNA, the ribosomes making proteins, putting them in a Golgi body, endoplasmic reticulum, then Golgi body, then out to the membrane. That whole process, that's how your cell controls. That's how DNA controls what the cell does. How does DNA control? We keep telling you that the DNA, the nucleus is the brains of the cell, right? We keep telling you that DNA controls. How does DNA control what the cell does? It does it by controlling what kinds of proteins it makes. That's the key. That's what's important. That's the most important thing. That will be tested all year long. All right. So now, transport substances that fit in the binding site, right? This is a carrier protein. Now, in a carrier protein, you actually have an actual site, a site that needs to bind to. So, with a channel protein, it's a specific size and shape that only allows one type of molecule through, right? Now, with this carrier, it's more like lock and key. In order for the key for this little substance, whatever this is, molecule, atom, ion, whatever. In order for this to get in, it needs to actually bind to a specific site. You see it? So now we call these carrier proteins. Those are proteins that bind to specific substances on one side of the membrane, and then what happens after it binds? The cell changes shape. The protein changes shape. When it binds, it changes shape. Remember the shape I kept pushing on you? I think a month and a half ago, I kept telling you structure and function, structure and function. I kept telling you the shape is so important. That proteins change shape. We talked about that. And so changing shape changes as the molecule, this thing binds to this part of the protein. Look at the change in shape. Look at it. Do you see the change in shape? Do you see it going from this, number 1, to this, number 2? And because it changed shape, what happens is that this molecule gets to go into the cell. There's no extra energy here. There's still no extra energy. It's still facilitating diffusion. You'll see when we need energy. I'm going to show you an example where you need energy. The classic, most famous of those energy requiring proteins is the ATP, sodium potassium pump. But we'll talk about that. Are we good with this? All right. So make sure, yeah. So these are the different kinds of this is facilitated diffusion here. So number 1, with facilitated diffusion, you have a molecule on the outside, right? This is a carrier protein. We're talking about carrier. And you see how it binds to the shape, by shape. Do you see that? You see, are you paying attention to the shapes matching? Almost like putting a square into a square hole, right? A square peg into a square hole, right? That makes sense, right? So here you have a round ball is going to a round opening, right? And when that binding, it's not a channel, right? It doesn't just go through. As soon as it binds, it changes shape. And as soon as it changes shape, this thing pops right back out. So it literally is just doing this. But it's only doing it because of the chemical bonding, the matching of the two molecules. There's no extra energy required.
Does it continuously do that?
Yeah, good question. And what happens after it leaves? This goes back to what? The open. It goes back, it changes shape again and goes back to the original shape and takes it another one. Just keeps doing that forever, as long as the protein lasts.
What if it's a different shape?
Then it doesn't fit and won't change.
So then
It just bounces right out. It only matches if the shape and qualities. It has to be the right charge, the right shape, etc. If it's the right quality of protein of substance, then the protein will change shape and allow it to go in. There are mimics. There are things that you could, What's a really good example? We talked about nerves early on in the year, right? We talked about a neuron, and we said that your brain, your memories are in brain cells, right? And that they're all connected. Now imagine if we had a substance that we could put into your blood that would stop the signal from one neuron to the other. What if we could stop this? What if we could take, let's say this was a neuron, what if we could do this, block it? What if something could bind to this and not let go? So the protein never changes shape again and doesn't allow anything else through. So all the signaling stopped. What would happen to you?
Can't you die?
You would die. You would die. Do you know what we call that? Neurotoxin. That's what nerve agent is. Something that blocks this process from happening. All right, so this is the end. Let's go ahead. We'll go back to all these. This entire presentation is already posted on the website. I will upload the video. It's on your website. I'll put it all down now. But let's go through the list. What are the words that we use today that you should have vocabulary cards, you should be practicing, you should be writing in your blue sheets once I print them? Number one.
Channel protein.
Channel proteins is an important one. I agree. Can I first say passive?
Passive transfer.
And another one on the other side of active. I think those are two really important ones. Active we haven't gotten to yet, but you should know what it is, right? In passive transport, we know you need what?
No, you don't need extra energy.
No extra energy, that's right. So there's definitions, they're in the notes. You guys can go back and look at that. But what's a vocabulary word that we might need? Certainly there's some background vocabulary, the idea of energy. Right, you should know that. So passive, right, good, gradient. So gradient, and it's a concentration gradient, right? Because all kinds of different gradients, but concentration. And what's another word you might need under passive that you might need to know? Anybody? Diffusion, I heard diffusion. Very good. Equilibrium. Maybe can I put dynamic equilibrium?
Yes. Is simple diffusion that important?
Yeah, absolutely.
Okay. Because we kind of, like, we went over it really kind of.
We spent 3 days last week going over simple diffusion.
Oh, that was simple.
That was simple diffusion. Diffusion, dynamic equilibrium, simple diffusion. So now diffusion, there's diffusion in general, the idea of diffusion, then there's simple diffusion,
Then there's
and then we have facilitated. If I misspell something or if you don't know how to spell it, just fix it later. Facilitated diffusion, and what's under facilitated diffusion? What are the two that are under facilitated diffusion? Channel proteins, channel proteins are one. So channel proteins, spelling's always been a weakness for me. Channel proteins, and what's the other one? Carrier proteins. So those are two kinds of facilitated diffusion you need to know. The more of this you study tonight, the better experience you're going to have tomorrow. Does that make sense? If you know this tonight, when you come in tomorrow and we're going over the material, you're like, oh yeah, I know that. Oh yeah, I know that. And of course, we better know that these two things here, we're talking about proteins, right? And that proteins are controlled by DNA, the production of proteins, a process. Simple diffusion happens with what? Here's a question that I would write. If I were trying to demonstrate in my notebook, because I was going to have my notebook graded, if I wanted to demonstrate that I was doing some revision, if I was wanting to demonstrate that I was doing some reflection, I would write questions like this. I would write questions, what determines simple diffusion? And I already told you the answer to that. What kinds of things determines it's the idea of simple diffusion? The concentration gradient. You have to have a gradient, right? That things move from one side to the other randomly without any extra energy. and high to low. And in a bilipid layer, when we're talking about a cell, what kinds of things cross, right? And what are those things that cross by diffusion, simple diffusion? Small and non-polar. And understand on your tests and on your quizzes and on your end of course exam, I promise you, it's not going to be what crosses the membrane, and you're not going to have to choose simple or small and non-polar. What they're going to do is give you molecules that they tell you are small or non-polar and ask you if it crosses the membrane. So you have to apply the concept. Do not expect that you're going to just memorize and then identify. That will not work. All right, I think we had a good day. Did we understand what we're doing? All right, so can we try to get here to energy tomorrow? Try to get some sleep tonight? All right, thank you.